This is how you set up your drawing page for exercise 3B, plant cell mitosis. So get your unruled paper and divide the page into 6 equal size sections as you can. And it's all caps, plant cell mitosis, underline it. And then just draw your vertical line from the line it's under all the way to the edge of the page. And then it's best to kind of eyeball it and put a little mark and then draw the horizontal lines. Usually if I just put a straight edge up and draw it without eyeballing it first, I'll have a lot of variation in the sizes. So you can pause it while you get that set up with 6 equal parts. When you get that done, and you come back and start it up again, and your 6 subheadings are your 6 procedures on that exercise. And in this order, from left to right and top to bottom. And I'll show you close-ups of that in just a minute. But that's when you see the other video, I gave you a crutch to remember in order. And Inez, pour me another tequila, dear. And so the I is interphase. The second one is prophase, metaphase, anaphase, telophase, and daughter cells. That's your 6 procedures in that order. Okay. As you get that set up, I'm going to zoom in on each one. And when you view the slides, this is what you'll be making your drawings for. I got to lean this back. And the CW, remember to spell everything out, that's the cell wall. And plant cells have a rigid cell wall due to the cellulose in it that we just referred to as being fiber. And so when you get those 6 rectangle shapes drawn, on every one of them, just go ahead and up close to the top or close to the bottom. I did mine at the top, label cell wall, but spell it all out. Just do that on all 6. And then when you look at the slide, there's going to be some pink grainy stuff around in the cell, and that's the cytoplasm. So you can go ahead and label that on all 6 down close to the bottom so it won't be in the way of your other labels. So do cell wall on all 6, cytoplasm on all 6. On interphase, you draw a nice big circle, and with the bracket label that nucleus. Now, when I had a chalkboard, I could shade things in. With these whiteboards and the markers, I can't shade. And so I just put a series of little dots. And if you can just do that, you might just shade it and then put little dots. It just looks grainy. You don't see any distinct structures in interphase. And the line that makes up the actual nucleus, and it's all bold-faced there in procedure 1. So the NM is nuclear membrane. Spell all these out. So the nucleus is the whole thing. The membranes is what surrounds it. And the chromosomes are so stretched out that they're not distinguishable. And so all of that material inside is called chromatin network. It's actually chromosomes, but you cannot distinguish them yet. So label that chromatin network. But do not draw any distinct lines in there. At best, just dots, but just shade it and kind of make it look grainy. And interphase means, inter means between, like intermission, interstate, intercontinental. Inter anything means between. So it's between divisions. And it may spend considerable time in that. And then when it's time, it starts undergoing mitosis. And then the first phase of active mitosis is prophase. And I gave you the term. It looks like a pile of worms. The chromosomes have kind of condensed, and now they're visible, so I'm going to zoom in on it. And the only other label you have are chromosomes. So let me zoom. I went the wrong way here. I'm going to zoom in, and you just put little short lines, but you notice there's no line around it. It's round shape, but the membrane that was around the nucleus has broken up, and all those little short things are chromosomes. It looks like a pile of worms. They're not in any kind of pattern. That's prophase. So you only have one additional label on that because you've already labeled cell wall and cytoplasm. And in your cells, you have 46 chromosomes, and in your pile, on your prophase, you see interphase, prophase, you'd have 92 chromosomes in that pile. And then all 92 in your cells are going to line up across the middle. And I gave you the crutch on the other video. On the metaphase, MET, they have met in the middle. So you can see there, they're all kind of lined up across the cell, kind of a zigzag looking line. And those dark structures, that's the chromosome. And then make those real bold. And then just barely, lightly with your pencil, draw some little filaments, but keep it very, very light. And they go to a point above the chromosomes and below them. And it's what the chromosomes travel on. And it's almost like the poles, the north and south pole. 46 of your chromosomes will go north and 46 will go south there. And that is metaphase. They have met in the middle of the cell. And then the next phase is anaphase. And the crutch I gave you was the chromosomes have pulled apart. So they pulled apart, and now they're all the way to the ends. And so those things that look like, if you hold your hand up with your fingers out, those are the chromosomes. And draw those real faint lines, but running between there. And the reason I labeled it with a dotted line, that you may or may not see them, those are the spindle figures. And it's bold-faced there in your lap. Whoops, sorry about that. So your joint needs to look similar to that. The chromosomes have to be long and spaced between them. That was anaphase. And next, I got to get my thing. I had a camera propped up here where I can get down. Got to take some more out. Hang on. Now I got to go back the other way. And I gave you the crutch on telophase as the chromosomes tighten. So you can see that they were long on anaphase with space between them. And now they're much shorter in kind of a corny crutch area that they have tightened. So those large mass with the CH, those are the chromosomes. And you see them at the top and at the bottom. And then draw those faint lines running between them. And you may actually be able to see those on the slide. They're more likely to show up on telophase than the others. And the CP, if you check your procedure, that stands for cell plate. It's not going to be on the one that is going to be on the slides that you look at, but it does develop later. It's just not on the one that I've shown you. And that will continue to get longer, and it starts growing until it goes all the way across, and it's going to become the cell wall between the new cells. So in telophase, and you've already put cell wall and cytoplasm on everything, and just label the chromosomes in one end, spindle figures, or is it fibers? It's spindle figure, and that is telophase. And when that cell plate grows all the way across the cell, the chromosomes break up. Now they become two brand new baby interphases. And on the other video, I stressed not to draw that dark purple thing that's in all of them. That's a nucleolus. Don't let that confuse you. And so can you tell that that looks almost like your interphase, except you've got two in the same amount of space you had one? And so you need to put all the labels you had on interphase on these, but not on both of them, as long as you put all of them total. So if you did cell wall on the top and the bottom before I drew that line, then all you have to add is the nuclear membrane label, the chromatin network, and the bracket for the nucleus. And that's the one you need to shade it. Try to make it look exactly the way that your interphase looked, maybe shaded in, but just kind of grainy looking. So I'm going to go back and go through all of them again. I got to tilt my bracket here, so I'll try not to wave it around too much. So this is in the order, and I'll give you the coiny crutch here. Inez, interphase. Por, prophase. Me, metaphase. Another, anaphase. Tequila, dear. If you drink too much tequila, you start seeing double. That's why I use Inez and tequila. If you drink too much tea, it doesn't make you see double. So you can pause this and go back, and this is the way you need to make your drawing for this exercise. And when you finish, it's going to look something like this. I'll be closing this off now. And that's the drawing you need to turn in, and you'll be looking at these actual slides. On the practical, you're not going to see my drawing. You're going to see the actual slides of the actual phases like you're going to be when you click on the plant cell mitosis. Okay, good luck.